Okay, can somebody please confirm you can hear me?
We can
We
hear
can
you.
hear you.
Thank you. Before we begin, is there any questions? Because are there any questions? All right. Let me say that I got one of the labs graded, and that lab, the second grading of it will be this Saturday. Let me see which ones they are. I'm opening up the grade book while I'm not showing my screen. The computer's a bit slow today. I got worksheet lab 3 graded, so that one will be, the second grading will be due this Saturday. Worksheet lab 4 is not graded, so it will not be due this Saturday, the second grading. Let me go ahead and put in the change for that one in there now. And that one, hopefully the second grading will be next Saturday. Come on, where do I edit that? Any questions about any of that? Remember, the second grading uses the until date. The first grading uses the due date. All right, got that change made for lab 4, and let's check lab 3 to make sure the until date's next Saturday. Yep, until October 18th. All right, any questions? If not, let me get out of the grade book and then I'll share my screen. Come on. I'm clicking the share button. It's not doing anything. There we go. Should be seeing my screen now. Is anyone getting into the Zoom meeting by scrolling down here and clicking on Zoom? Because I'm not getting this link to work. It does open the page, but then when I click on the link in the link in the page doesn't work.
I did that today and it worked.
It is working for you guys. Good. It's not working for me, and I don't know why. So I have to log into www.zoom.com and then log into my account. And I have to log in for my password and username for Zoom, and then it asks for my password and username for clark.edu, and then it then has to verify my existence by sending a message to my phone. And if you have Okta, it uses Okta. Anyways, that's what's happening with me. Why I was late with the first lab, but I knew it was happening, so in the lecture I got it done earlier. All right, I have that we were talking about the prokaryote unusual shapes last time, and we stated that most bacteria are cocci, bacilli, or spirals, but there are a few exceptions. And then most bacteria are monomorphic in their shape, but there are a few species which are pleomorphic. Let's talk about the cellular growth arrangement, also called the cellular arrangement. This is how the cells stick together, what type of arrangements the cells have when they stick together. First, which isn't shown here, let me blow this up. You have singlets, like right there is a singlet. So you can have single cocci or single bacilli, and we call those singlets, but you can call it single cocci if you wish. There's also diplococci, and that's when two cocci stick together. You can have streptococci, and this is when the cocci are stuck together in a line. You can have tetrads, and that's when four cocci are stuck together. You can have sarxinia when eight cocci are stuck together. And you can see seven here. The eighth one is hidden right under there. Oops, didn't mean to do that. Trying to show this, bring that up there. You can also have staphylococci. Staphylococci literally means ball shaped cells, meaning the cocci, growing together as a clump of grapes. If you were to put a stem on this right here, that kind of looks like a clump of grapes. And those are the arrangements that the cocci can have. The arrangements do occur because of the plane of cell division. You don't really need to know the plane of cell division because I won't test you on it, but the reason why diplococci happen is because the plane of division is this way, and streptococci also has this plane of division. The difference is that all the cells stick together, whereas the tetrad has two planes of division, Sarxinia three planes of division, and Staphylococci I have no idea. For bacilli, they do have cellular growth arrangements also, but they have fewer. They have a single bacillus or a singlet. Diplobacilli, that's two bacilli stuck together on the short ends, making a long string. Streptobacilli also stuck together on the long short end, so we make a very long string. And this reminds me of the old sausage links. I don't know if you guys ever saw those, but they used to make sausages together like that. Coccobacillus, as I mentioned before, is not a cellular growth arrangement. It should not be in this slide because it's talking about cellular growth arrangement. A coccobacillus is a cell shape, not a cell growth arrangement. And then you can also have bacilli stuck together on the long end, making a palisade, which is what the name is. It comes from the palisade fence. Let me see if I can get paint to work. No, that didn't work. See if I can get it to work here. There we go. In the old days, like if you read Mark Twain, he was, well, it's Mark Twain, I think it's Tom Sawyer who was painting the palisade fence. And what you do is you just put a board, sorry, not very straight, but you get the picture. And you stack them this way, lengthwise. And then there's the ground at the bottom. I have no idea how they're stuck together, if they're nailed together or what, but that's a palisade fence, and then the ground's down here. And that sort of looks like the palisade of the cells, why it's called the palisade. Anyone remember that from Tom Sawyer and Huckleberry Finn? Um, I think it was Tom Sawyer who was painting the fence, and he was trying to convince Huckleberry, was it Huckleberry Hound? I don't remember his name. Huckleberry that it was fun painting the fence, and Huckleberry should paint the fence for him. Anyone remember that? Oh
I
well,
do. It's
good.
Huck Finn.
I do too.
Yeah, it's Huck Finn, okay. When we're talking about the cellular growth arrangements, you should not confuse the family Bacillus with the cell type Bacillus. If it's lowercase b, then that's the cell shape. If it's uppercase B, then that is the family of Bacillus. There's also the family of Streptococcus, like Streptococcus pyogenes, and that you should not confuse with Streptococci, the cellular growth arrangement. Streptococci. And that one is spelled differently. Cocci is the Streptococci is the cellular growth arrangement. Streptococcus is the family. All right. Any question about the cellular growth arrangement? If not, let's talk about some of the structures of prokaryotic cell. This slide is showing you the structure of a typical prokaryotic cell, naming them. All the ones in red are found in all prokaryotic cells. The cytoplasm, the plasma membrane, and the DNA here, those are actually found in all cells. The ribosomes are found in all cells, but the ribosomes of a prokaryote cell are smaller than the ribosomes of a eukaryote cell. And then the DNA of a prokaryote cell is found in the nucleoid region. You remember the DNA of a eukaryote cell is found in the, sorry, I'm drawing a blank here, the nucleus. The nucleus has a defined boundary between the cytoplasm and the nuclear protoplasm, the, what is that called? The nucleoplasm. And that's the nuclear cell membrane, making a boundary between the cytoplasm and the nuclear protoplasm of the nucleoplasm. However, in a prokaryote cell, there is no boundary. The nucleoid region is wherever the DNA is floating in the cytoplasm. So the DNA is actually, most of the time, it's dissolved in the protoplasm of the cell, the cytoplasm, and wherever that DNA is floating, that is the nucleoid region. Wherever it's not found, it's not the nucleoid region and it's external to the nucleoid region. But my point is there's no boundary between the nucleoid region and the cytoplasm. It's wherever the DNA floats, that's the nucleoid region. The nucleoid region does tend to stain differently when we apply dyes, or even in the electron microscopic image, it tends to stain a little different. It's just because DNA is a long and unusual molecule. It makes it stain a little different. All right, I think I've talked about the cytoplasm, the plasma membrane, also called the cell membrane, and the DNA are in all cells. You can have ribosomes in all cells, but you should know the ribosome of a prokaryote cell is smaller than the ribosome of a eukaryote cell. Some other regions of the cell, there's the pilus, also called the sex pilus. We'll talk about that when we talk about genetics in chapter 8. There's a capsule on some prokaryote cells. Capsules are even found on some eukaryote cells, but in a prokaryote cell, if the glycocalyx can be seen in the light microscope and has lots of molecules in it other than water, and is firmly attached to the cell wall, then that is the capsule. It's something external to the cell, something the cell secretes, and it's considered outside the cell because it's outside the cell wall. So the cell wall is the last structure, if you wish, to a cell. It is not the structure, though, that determines what comes in and out of the cell. That's the cell membrane. But the cell wall is considered a part of the cell. And the capsule is external to that, so it is considered external to the cell. The cell wall is the next part, and I already talked about that. The cell wall gives this cell support, shape. If the cell is cockeye, it's because the cell wall is a cockeye. Or coccus, I guess. And if the cell is a bacilli, it's because the cell wall is a bacilli shape. Plasmids are a region of DNA external to the chromosome. A plasmid is a small piece of DNA, usually having only a couple of genes, maybe here's a dozen or two handfuls, whatever you want to call it, at most, usually. And a chromosome is a large piece of DNA which has proteins in it, but we'll talk about that later. Um, the important part is a chromosome has thousands, perhaps tens of thousands of genes on it. Any question about that? So the main difference between a plasmid and the chromosome is the chromosome is much bigger and has many, many more genes on it than a plasmid. Plasmid is external to the chromosome, and the plasmid is just a molecule in the cytoplasm. Another part of some cells is the flagella. If the cell can swim or move by locomotion, and it's a prokaryote, then it has at least one flagellum, if not multiple flagella. And that's because the flagella is the only structure in a prokaryote cell that allows the cell to move. It's different from a eukaryote cell, which has three structures, and I think I've talked about them and we'll talk about them later. Something seen on some cells are fimbriae. They are hair-like structures attached to the cell membrane or the cell wall, sticking out from the cell. So most of the fimbriae are external to the cell, outside the cell, but some of it is attached to the cell membrane or the cell wall. Oh, one more thing I need to talk about, an inclusion, also called an inclusion body. An inclusion is molecules, usually of one type, that are being accumulated by the cell. And so much molecules form in the cell that they begin to precipitate out of solution. If they do precipitate out of solution, we then call it an inclusion. So an inclusion is something the cell is storing in vast numbers of molecules, and there's so many of them that they actually precipitate out of solution. And then there's this solid structure in the liquid cytoplasm, and that we call an inclusion. And there you can see one there and one there and one there. Any question about all of these structures? All right. As I mentioned, the glycocalyx is secreted by some prokaryotes, not all. It's outside the cell wall. It's usually sticky. And there are three types of glycocalyxes. And you can call that glycocalyx, glycocalyx. Both pronunciations are correct. The first type of a glycocalyx is a capsule. It's neatly organized. You can see it in the light microscope. It has many molecules that are not water, meaning other than water, and it's attached to the cell wall. The capsule can prevent phagocytosis by a white blood cell or another immune system cell. So the capsule tends to protect the bacteria from our host immune system. The second type of a glycocalyx is called the slime layer. It's an unorganized, loosely attached structure that's mostly water. You cannot see the slime layer under the light microscope. You have to use an electron microscope to actually see the slime layer. Any question about any of that? And then the third type of a glycocalyx is the extracellular polysaccharide. This is the stuff that makes up biofilms. And what happens is you have cells that start making a capsule, and then you have another cell over here making a capsule, and then eventually the capsules come together, and then a whole bunch of cells are doing this, and they all start coming together, and that forms the biofilm. Cells that form a biofilm are harder to treat than cells that do not have a biofilm. And for now, that's all you need to know about biofilm. We will talk a little bit more about biofilms in a later lesson, but in this class, we're not really going to talk much about a biofilm. I can state that if you have a bacterial infection that has a biofilm, they are harder to treat than an infection that lacks a biofilm. Any question about any of that? All right. The flagella is the structure that a prokaryote has for locomotion. It is mostly outside the cell wall, like all of this portion here you're seeing and this portion here. It's outside the cell wall, but you should realize that there's a small portion of the flagella which is inside the cell wall. So here's the cell wall there. And then there's even a small portion of the flagella which is inside the cell membrane. The flagella are made of proteins called flagellin. So flagella are made of the protein flagellin. Any question about any of that? There are four basic arrangements of bacteria flagella. You can have monotrichous, and that's one flagella on one end. You can have amphitrichous, that's one flagella on both ends. You can have lophotrichous, multiple flagella on both ends. And then you can have peritrichous, and that's multiple flagella all over the cell. Remember that if you're looking at the flagella arrangement, you need to count the highest number that the species has. So if the highest number is 0, you say there's 0 flagella in this species. But if there's 1, you say that the flagella is monotrichous. If there's 2 on each end, you say that the flagella is amphitrichous. If there's several on both ends, you say the flagella species is lophotrichous. And if there's multiple flagella all over the cells, you say it's peritrichous. Now, because the flagella need to be made by the cell, and when the cell first divides, the cell will lack any flagella. So you will have some cells right after cell division that lack the flagella, and then they can start making flagella, and they'll start with the monotrichous, If the cell only species can only make monotrichous, we say that species is monotrichous. But if it can make higher than monotrichous, it'll then become amphitrichous, and then higher than that will become lophotrichous, and then higher than that will be peritrichous. As was discussed in the lab, whenever you're giving the flagella arrangement, you should report it as the highest number of flagella that you can see. Any question about any of that? All right. So if cells are motile, meaning they rotate or they run or they swim, it's because the cell has, if it's a prokaryote, it's because the cell has a flagellum or a flagella. If I haven't mentioned, flagellum is a singular, flagella is a plural for that word. Any question about any of that? So if cells can swim or rotate or tumble, they have at least one flagellum. Flagella are important, at least for prokaryote cells, because of taxis. Taxis is the movement toward or away from a stimuli. So let's play, you were to put a toxic metal substance up here, the flagella will move away from the toxic substance, and then they move down. They would actually move by chemotaxis. Chemotaxis is the conveying the signal message from the cell to the nucleus by chemotaxis. And then the nucleus or nucleoid region, if we're a prokaryote cell, will then have the flagella beat in the opposite direction. If it uses the same flagella, it may, if it has multiple flagella, it may just use one on the other end and whip it to make the cell go this way. Any question about any of that? So taxis is the movement toward or away from stimuli. You can have chemotaxis, as already discussed, in which case the signal message comes from a chemical. Or you can have phototaxis, and that is movement towards or away from light, especially sunlight. And here, these cells must not be photosynthetic because the light source is here, but yet the cells are swimming this way. Any question about any of that? Excuse me here. It feels like I'm going to sneeze and trying to prevent that. We'll see if I succeeded. Next structure I want to talk about is the endoflagella, also called the axial filaments. I call it an endoflagella because it is a flagella, but it's actually inside the cell. The endoflagella are only found in spirochetes, and one end of the endoflagella is anchored inside the cell wall, and then the other end wraps around the cell like that. Let me blow this slide up. So it wraps around the cell like that, or like this. And if you do the cross section, let me blow this up so you can see it better. You can see that the axial filaments, also called the endoflagella, are actually inside the pink cell wall. Any question about any of that? So the endoflagella are inside the cell wall. And it is true, the cell wall is the structure here, and then the cell wall bends around the endoflagella. the endoflagella move by, if I get this to play, there we go, move in a characteristic direction that we call corkscrew-like movement.
Grammarly now uses AI to help you write, not just revise. Type a prompt and generate a persuasive.
Sorry about that. All right, let's start over. So as you can see, the cell is going to rotate that way and move this way. And it can change directions, moving that way. You just change the way that you're moving, or which way the cell is rotating. You can change direction. The spirocade is also flexible enough that it can fold up itself and then start moving instead of north to south, east to west, as shown right here. And that tells you how flexible the cell wall of a spirocade is. Much more flexible than the spirals. Any question about any of that? It's thought that this corkscrew-like movement can actually allow at least certain cells to penetrate the mucous membranes as well as penetrate patient's tissues. For example, it's thought that the treponema pallidum, or is it treponema pallidum, the bacteria that causes syphilis, is a spirochete. And it's thought that why the spirochete can get through the mucous membrane and then get through the patient's tissues is because of its corkscrew-like movement, allowing it to burrow through the host tissue or the mucous membrane and then through the cells. We'll just word it that way. Any question about any of that? All right. Fimbriae are shorter, straighter, and thinner and hair-like, and they're straighter, shorter, and thinner than the flagella. The fimbriae are not used for movement. They are used for attachment. So they attach to some surface. They do not allow the cell to swim or move. The fimbriae may be why the prokaryote cell or the bacteria is allowed to attach with you and then infect you. It has to get through the host defenses, but once it does that, it can infect you. So the fimbriae are used for attaching to humans, and oftentimes the receptor, or is it the ligand? The ligand on the bacteria, if it has fimbriae, the attachment molecules of the bacteria will usually be on the tips of the uh, fimbriae. Any question about any of that? All right. The pili, also called the sex pili, is a hollow structure that is made by this cell. And you can see, let me blow that up. You can see fimbriae on this cell, but the fimbriae are shorter and thinner than the pilus. The pilus's job is to go out and find a gram-negative cell and then skewer it with the sex pilus. The sex pilus can then act as a winch to bring the cells together. The purpose of attaching the cells together is that it allows a plasmid from this cell that has the pili to move that plasmid into this cell. And it replicates before it moves, so this cell is a replica of the plasmid or the viral piece of DNA. We'll talk a little bit about that later, but for now, let's just call it a plasmid in the prokaryote cell. And then that plasmid copies itself, and then one copy is sent through the sex pilus into this cell, which lacks that plasmid. It also lacks the gene to become to make a sex pili. Any question about any of that? All right. The reason why this does not need to bring the cells really close together, although I think in the next show, nope, I don't. That doesn't hurt bringing the cells really close together. And then literally the cytoplasm can flow from this cell into that cell and vice versa. But the important part is that the plasmid that codes for the sex pilus, by the way, is the plasmid that replicates and then sends a copy to this cell here. How? Well, that sex pilus is actually hollow. Any question about any of that? All right, let's talk a little bit more about the prokaryote cell wall. And specifically, for most of this lesson, we're going to talk about the cell wall to bacteria. The cell wall of archaea are different. And as you know, the cell wall of bacteria and archaea, for that matter, are different from the cell wall of a eukaryote cell. Humans are eukaryotes, but humans are animals, and animals do not have a cell wall. Any question about any of that? All right, the cell wall of a bacteria is complex, semi-rigid, responsible for giving the cell its cell shape. The cell wall protects the cell and prevents osmotic lysis. For example, if a patient has an infection with a bacteria, that bacteria is going to have so much molecules in it that the molecules will want to leave the cell, even if it's a nutrient like glucose, will want to leave the cell and go into the lumen of the intestine or wherever you are, like the blood, for example. Um, so the protein will tend to leave the cell if it's at higher concentration inside the cell than outside the cell. So it doesn't make any sense for the cell at this point to make ATP and use a transport protein to bring the molecule that the cell wants inside the cell. Any question about any of that? Most molecules, they'll come into the cell by binding to a receptor, and then the molecule will be brought into the cell. A very large protein, if not any protein, I'm not sure, but most proteins, they're too large to come through the cell membrane. So how does the cell bring a molecule in that it wants? Well, it brings it in with an ATP synthase molecule, if it has one. I mention that because there are some cells that lack a cell wall. There are also other cells where the cell wall may be destroyed, such as a cell almost killed off by antibiotics. Oh, I was starting to say, if a patient has a bacterial infection, The bacteria tend to have more water outside of them than water inside of them. And the reason why the bacteria has less water inside the cell than your eukaryote tissue is because the cell will accumulate the molecules it needs and then start storing them. So on all of the Jupiter 2 missions on Lost in Space, for example, the what was I going to talk about? And how is it relating to the cell wall? The Jupiter 2 would be storing stuff inside it, sort of like the prokaryote cell stores things inside of it. And so many molecules are stored inside the cell wall that there is more water outside the cell than water inside the cell. And because of that, the cell wall will protect the cell from osmotic lysis. And if you dump a bacteria that has an intact cell wall into a beaker of water, the cell wall will prevent osmotic lysis. And we'll talk a little bit more about that, I think, in this lesson when we're talking about the cell membrane. The cell wall is located outside the cell membrane. It does contribute to the pathogenicity of some species of bacteria. For example, any gram-negative cell that has a cell wall will contain a toxin. It's called endotoxin, and it's a toxin inside the cell wall of a bacteria. Any question about any of that? And obviously, if the bacteria has a toxin, that contributes to the pathogenicity of that species. The cell wall of bacteria is made by peptidoglycan, meaning peptidoglycan is the main molecule in the cell wall of bacteria. Peptidoglycan is itself made up of repeating disaccharide, NAG and NAM, and then it's the links of NAG and NAM are linked together by short polypeptides. So here we have a cell wall, and you can see NAM, let's go here, NAM followed by NAG, followed by NAM, followed by NAG. So it just repeats this disaccharide. Remember, a saccharide is a simple sugar, and NAG and NAM together are a disaccharide. And they're just hooked together in a link using a chemical covalent bond. And then the links repeat one after the other in a plane. And then we have multiple planes, like there's the second plane underneath the first plane. There's the third plane underneath the second plane. Holding the peptidoglycan lattice together, meaning the NAG and NAM lattice together, are short peptides. Here we have a short peptide linking this NAG and NAM chain, come on mouse, to this NAG and NAM chain, and there's the short peptide there. We also have a short peptide hooked to this short peptide there, running to this short peptide there, and this short peptide is linking that NAG and NAM chain to that NAG and NAM chain. So this short peptide then links this NAG and NAM together. Now, I don't care what you call them, but NAG and NAM are easy names, and NAG stands for N-acetylglucosamine, and NAM for N-acetylmuramic acid. Both the names and the initials probably mean about the same to you, meaning I doubt you know what NAG and NAM are, and my point is you can name them either NAG and NAM or N-acetylglucosamine and N-acetylmuramic acid. The short peptides linking the NAG and NAM together are actually how penicillin damages the bacteria cell wall. Penicillin will get in right here, excuse me, and if this link has not been made yet, penicillin will prevent that link from being made, meaning penicillin, at least penicillin G, some of the other simple penicillins, they need to work on a growing cell, a cell that is actively putting down its cell wall. If they were to be used against a cell that is not actively growing, and the cell is, let's say, it's hibernating or in a stasis of no metabolic activity, then penicillin G is not likely to kill the cell, even if the penicillin G can get through the cell wall. The point is that NAG and NAM, if it is not connected by this short peptide there, that'll make a gap in the cell wall. If there's a gap or a hole in the cell wall, the cell membrane can protrude out of that hole. I don't know if you can see it. And when the cell takes in more water, that protrusion will gain in size. And that protrusion will continue to grow until it pops the cell membrane, and then the cytoplasm will leak out of the cell, and that will kill the cell. Any question about any of that? And how the penicillin G is actually acting is it's stopping this peptide link from happening, creating a gap in the cell wall. And then the cell relies on, or I guess the clinician is relying on water to move into the bacteria cell. Most of the time that works because the cell is metabolizing things. It will use up those molecules that are stored in the cell. But like I said, if the cell is just in stasis, not growing, not dividing, not doing much actually, very low metabolism in a cell that's not dividing or not growing. Trying to think where I was going with that. So penicillin G damages the cell by forming, getting in between this NAG and NAM chain and preventing that cross-link of the peptide from forming, creating a hole in the membrane, and then the water coming into the cell will eventually kill that cell. And that's how penicillin G actually works, which tells you that when you have a bacterial infection, most of the time the bacteria will have less water than your tissues. And that's why penicillin G tends to work, at least on gram-positives. Any question about any of that? All right. Well, this structure here, even though it looks impressive and it can be 30 to 100 layers or more on a gram-positive cell, it does not really prevent much from entering the cell. Any molecule that is water soluble can get right through the cell wall. And that's actually how we get our cells to obtain their sugar and their salts and their magnesium and chloride and almost any other ion the cell needs or any molecule the cell needs. All right, any questions about any of that? So remember, the gram-positive bacterial cell wall differs from the gram-negative bacterial cell wall. The gram-positive cell wall has a thick peptidoglycan layer, usually 30 to over 100 layers of peptidoglycan. This is only showing you 3, and you have to remember it's more than 3. It's actually 10 times more. The minimum is around 30 layers of peptidoglycan in the gram-positive bacteria cell. And there can be many, over 100. You can note the gram-positive bacterial cell wall, besides having peptidoglycan as the main molecule, hooked together by the lattice of peptides, short peptides. It does have a few other molecules like teichoic acid and lipoteichoic acid. Lipoteichoic acid is the same as teichoic acid, except lipoteichoic acid has a lipid layer attached to it, and that lipoteichoic acid actually anchors itself in the lipid bilayer of the cell membrane. All right, any questions about the cell? Let me state that you can have proteins spanning, meaning going through the cell membrane, and you can have proteins inside the cell wall as also. I probably shouldn't have ended that because I got other, in a minute I'll have other slides which I have to blow it up, but all right, so the gram-positive cell does have teichoic acid, 2 types, lipoteichoic acid, and then teichoic acid, which is called wall teichoic acid. It's thought that the lipoteichoic acid can help regulate the movement of cations, meaning a positively charged ion can move down the teichoic acid and then wait for entry inside the cell membrane. If teichoic acids, and I'm saying if because at the writing of our textbook it wasn't known, if teichoic acid does help regulate the movement of cations through the cell wall, then you should know that the regulation of tomatoes and carrots and beets that I had for my lunch, what was I thinking here now? Sorry, I did it again. Well, the teichoic acid can help regulate the movement of cations, but it's not necessary because a cation or any ion, negative or positively charged, can be dissolved in water, and then any molecule dissolved in water can go right through the gram-positive cell wall. It is true that the cations do come down the teichoic acid, meaning the teichoic acid can help the cation come through the cell wall, but it isn't necessary. You don't need the teichoic acid because the ion or any cation can actually move right through the cell wall. The polysaccharides found in the gram-positive cell wall can give antigenic variation to the cell. Like O111, that is an antigen from a polysaccharide in the cell wall of the bacteria. And I guess the one I'm talking about is E. coli O111. That is antigenic variation from polysaccharides in the cell wall. All right, the gram-negative bacterial cell wall is fundamentally different than the gram-positive cell wall. Once again, we have a thin bilayer, lipid bilayer. We call that the cell membrane. We have a thin layer of peptidoglycan. And I said layer, and this is showing you only one layer, but in reality, the cell wall is a minimum of 3 layers of peptidoglycan up to about 20 layers of peptidoglycan. So it's almost always more than one layer of peptidoglycan. It's in a gram-negative cell, it's between 3 and 20 most of the time. All right. Let's start down here. Once again, let's start. Yeah, let's say that the cell wall can be divided into 2 parts. There's the peptidoglycan layer of the cell wall, and then there's a lipid bilayer that is called the outer membrane of the cell wall, and that's shown there. The outer membrane of the cell wall is very similar to the cell membrane. In fact, the innermost layer of the outer membrane of the cell wall, this one here, is mostly phospholipids. You can find some proteins in it, some carbohydrates, probably some lipids as well. But most of the molecules of the outer membrane of the cell wall, gram-negative cell wall, I should say, contain phospholipid, which for this layer of the lipid bilayer, that's the main molecule. And there's only a few that are not phospholipid. Let me blow that up a little bit and then you can see the phospholipid. There's a phospholipid, and that's also the main molecule in the membrane that we call the cell membrane. The outermost bilayer, meaning the upper bilayer, does have phospholipids in it, and it is the main molecule, but it does have a significant numbers of other molecules in it, including lipid A, and then an O polysaccharide molecule attached to that lipid A. Lipid A is the important molecule in the gram-negative cell wall because lipid A is an endotoxin, something that is toxic to us. And so if you get a gram-negative infection, or you have a patient with a gram-negative infection, you should realize that this patient has an infection, member of Staphylococcus family, causing the skin disease, and that the gram-negative cells will have an outer membrane containing lipid A, and so that patient will be exposed to lipid A, which is an endotoxin. And what's the clinician to do? If you've got a patient with a gram-negative infection, well, they want to get rid of the infection, so they do give the anti-penicillin drugs. But like I said, you should realize that gram-negative infection, as long as it has a cell wall, it'll have lipid A, which is a toxin, and the patient's just going to have to deal with it. Hopefully they have enough room to maneuver, because if the patient can't deal with it, the endotoxin will eventually kill them. All right, one other molecule I need to talk about that is in the cell wall of a gram-negative cell, and that's the porin protein. The porin protein is found in the outer membrane of the cell wall. The porin protein allows many molecules that the cell wants to enter the outer membrane of the cell wall. So you've got the molecule halfway into the cell now. There is no porin protein in the cell membrane. What we have for bringing molecules into the cell membrane is a transport protein. And the difference is the porin molecule allows many molecules to enter the outer membrane of the cell wall. And then when the molecule reaches the cell membrane, the only way to bring the molecule in is by a transport protein. And the difference between the transport protein and a porin protein, the porin protein allows many molecules to enter. The transport protein only allows one molecule to enter. Usually the transport protein is pumping against its concentration gradient, not always, but often. And it wants that molecule, so it's going to pump it into the cell. But before the molecule can get in this region of the gram-negative cell where the ATP ACE enzyme is, the molecule has to move through the outer membrane of a gram-negative cell. And if it can, it'll move through the porin protein. Not all molecules go through the porin protein. For example, penicillin G cannot do damage to a gram-negative cell, even though it kills a bunch of green leaves or branches, whatever you want to call them, fir needles. Anyways, the cell wants the molecule. It has to come through the porin protein. The porin protein allows many molecules to enter the cell. But to actually enter through the cell membrane, you need a active transport protein, or at least a transport protein, to bring the molecule inside the cell. And generally, the active transport protein is specific for only moving one molecule. Like we have two different active transport proteins on the cell. If you want to move in both the sugar, glucose, and fructose, you need two proteins because each will recognize only their own molecule, at least if it's an active transport protein. Foreign protein is different, and that is it allows many molecules to enter the cell, or at least the cell membrane, or the outer membrane of the, let's call it the outer membrane of the cell wall. All right, penicillin G can go right through the gram-positive cell wall, so it can do damage to the cell wall, assuming the cell is actively putting out its cell wall. Meaning you can't have a cell in stasis and give it penicillin G because the cell concentration won't be high enough to kill off the bacteria causing the infection. Hopefully I said that right. All right, any question about the gram-negative versus the gram-positive cell wall? So the gram-negative outer membrane does have some different molecules, including lipopolysaccharides. That's the lipid A with the polysaccharide tail attached to it. Lipoproteins, excuse me, phospholipids. These molecules are not found in a gram-positive cell. There is the periplasm. I don't really talk much about it. The periplasm is the region of the cell wall of a gram-negative cell where the peptidoglycan layer is. So the periplasm is in between the cell membrane and the outer membrane of the cell wall. The gram-negative outer membrane can offer the cell protection from certain complement proteins, from certain antibiotics. For example, penicillin G cannot get through the porin protein, and if it can't get through the outer membrane of the cell wall through the porin protein, it's not going to damage the peptidoglycan layer by getting in between the NAG and NAM chains and preventing the crosslinks from happening, meaning crosslinks of the short polypeptide chains. The O polysaccharide, meaning the carbohydrate attached to lipid A, can provide antigenic differences. For example, the phagoside, the E. coli O157, that O is an antigen of E. coli that differs from the main E. coli, which we don't even give it numbers after it. We just call it E. coli. And that O157 is an antigen coming from the bacteria, coming from that hair, the old polysaccharide, attached to the lipid A. And we talked about lipid A being an endotoxin and porin molecules. Any question about any of this? All right, so the gram-negative cell is a little more protected than the gram-positive cell because it has a outer membrane, and any molecule damaging the cell has to get through that outer membrane. Penicillin G cannot get through that outer membrane. That doesn't mean we don't have antibiotics to work against a gram-negative cell. For example, ampicillin, which is, I think, the third member of the penicillin family found, meaning the third member of the family found, does allow the molecule of ampicillin, the antibiotic, to move through the porin protein and get through the outer membrane, and then it damages the cell wall in the same way penicillin G damages the cell wall of a gram-positive cell. All right, any questions about this slide? All right, let's take a look at the gram-positive cell wall. It has a thick peptidoglycan layer. The thick peptidoglycan layer is rigid. The gram-negative cell wall has a thin peptidoglycan layer, and its thinner layer is rigid, but is not as rigid as the gram-positive cell. Teichoic acid is a molecule found in the cell wall of a gram-positive cell. There's no teichoic acid in the gram-negative cell wall. There's no outer membrane of the cell wall in a gram-positive cell. There is an outer membrane in the gram-negative cell, and it's important because lipid A is found in that outer membrane, and lipid A is something that is toxic to humans, or is an endotoxin which is toxic to humans. Any question about any of that? All right, let's talk a little bit about the Gram stain mechanism. You already know that crystal violet iodine crystals form a complex between the crystal violet and the iodine, which tends to stain the cell. Gram-positive cells, this crystal violet iodine complex does not leave the thick peptidoglycan layer. So the molecule is big enough that it does not leave the thick peptidoglycan layer as long as you apply the decolorizing agent for the correct amount of time. And then on the gram-negative cells, the crystal violet iodine complex does form in the peptidoglycan layer of the cell wall, but the alcohol acetone wash will dissolve the outer membrane, so it disappears, of this of the cell wall. And then the crystal violet iodine can wash out of the thinner peptidoglycan layer. And that's why the gram-negative cells do not stain purple, but the gram-positive cells do. And then we apply the dye safranin after decolorizing the gram-negative cells, just to give the cells a pink color, and then it makes it easier for us viewing it under the light microscope. Any question about any of that? All right, here's a good table summarizing the cell walls between a gram-positive and gram-negative cell. You only need to know the first part of the table. You can ignore the later part of the table. So I guess the second column, the first column is characteristic we're going to look at. The second column is the gram-positive cells. The third column is the gram-negative cells. So in the gram reaction, you retain the crystal violet iodine complex, so the cells remain purple. In the gram-negative cells, the crystal violet iodine complex washes out in the decolorizing step, and then we stain the cells with safranin, making the cells look pink. Peptidoglycan layer is thick and multi-layered in the gram-positive cells. This table is got an error in it, or perhaps it's so old that it wasn't known, but the peptidoglycan layer is thinner than in the gram-positive layer, but it's not a single layer. It's a minimum of 3 layers of peptidoglycan, and it could be up to 20 layers of peptidoglycan, one on top of the other. It is much thinner than the gram-positive cells. Teichoic acid present in gram-positive cells, absent in gram-negative cells. You don't need to know about the periplasmic space, but the outer membrane is absent in the gram-positive cells. It's present in the gram-negative cells. And then lipopolysaccharide, which will contain lipid A, which is the endotoxin. There's none in the gram-positive cells, and then it's actually present in high amounts in the gram-negative cells. Any question about any of that? All right, so we've talked about some cell walls. Now let's talk about some atypical cell walls. The first, and I should move this up, is the archaea. Everything we've spoken about has been true for the bacteria, but actually none of it, or almost none of it, is true for the archaea. The archaea do tend to have cell walls. It's true that some archaea lack cell walls, but some bacteria lack cell walls also. If the archaea have a cell wall, it is not made of peptidoglycan. It is made of pseudomurine, and that is actually another disaccharide that is found in the cell wall of a archaea. And that just makes the archaea different than the bacteria, which is not surprising because it's a different domain. Some other differences which happen in the domain bacteria is with the family Mycoplasma and the family Mycobacteria. The family Mycoplasma is a family in the bacteria, but these bacteria differ because they do not have a cell wall. They only have a cell membrane, much like humans and animals, and they don't make cholesterol to put in their plasma membrane the way animals do, but they put in a sterol in their cell membrane to help give the plasma membrane or the cell membrane strength and support. And that's much the purpose of having cholesterol in the cell membrane of animal cells? Any question about any of that? So the mycoplasma differ because they lack a cell wall. That tends to make the cells, by the way, pleomorphic if the cell lacks a cell wall. All right, let's move to the Mycobacterium, another family of bacteria that differ in their makeup of their cell wall. The mycobacteria are said to be acid fast staining cells because they have a lot of mycolic acid in their cell wall. Mycolic acid is really only found in two families of bacteria, the highest concentration being in the family Mycobacterium. But mycolic acid is in the cell wall, and that mycolic acid prevents water as well as any molecule dissolved in water from entering the cell or leaving the cell. So the mycobacterium can stay alive longer than almost any other bacteria in a harsh environment like a dry desert environment. The mycobacterium can stay alive because all that mycolic acid in the cell wall helps prevent the cell from losing water. Any question about any of that? And then that mycolic acid also allows the cells to be acid fast staining positive, where the acid fast stain gets in the cells and then it doesn't wash out. All right, any question about any of that? If not, I think, yeah, we're not very far ahead. Let's keep moving. See if it goes. Oh, there we go. You can damage the prokaryote cell wall, damage the prokaryotic cell wall, either get rid of the cell wall or putting holes in it, and then that can make the cell sensitive to osmotic lysis. You can treat the cell wall with an enzyme, lysozyme, and that will digest the disaccharide in peptidoglycan. You can also treat the cell with penicillin, like penicillin G, and that will damage the cell wall, putting holes in it. And then water, when it moves into the cell, will eventually burst the cell membrane. You don't need to know the terms for a wall-less gram-positive cell or a wall-less gram-negative cell, but you should know we can damage the cell wall. All right, gram-negative bacteria are not as susceptible to gram-positives to penicillin G due to the outer membrane of the cell wall. Gram-negative bacteria are also susceptible to antibiotics that can penetrate the outer membrane, such as the antibiotic streptomycin. Streptomycin is a fat or lipid-soluble molecule, and it can go right through the lipid bilayer of the outer membrane of the cell wall. And then I mentioned that ampicillin can also go through the outer membrane of the gram-negative cell wall. So both of these antibiotics can kill a gram-negative cell. All right, any questions about any of that? If not, I am going to end it here because I don't want to talk about the plasma membrane. We don't have enough time. Let me state there is no lab today. I will be logging in for the first 15 minutes of the lab time to answer questions, but there is no lab today. All right, any questions? All right.